Bismillahirrahmanirrahim. We start the video 2 for this question which we've done with this paper which we've done in a previous video, and we continue with the questions that are left of June 19, paper 1-3. Now we start with question 23 because we've covered question 1 and 22 in the first video. This is the second video on this paper. So question 23, the diagram shows a cell during mitosis. So definitely this cannot be a bacteria because bacteria does not undergo mitosis, so it cannot be a prokaryote. Nor does virus undergo mitosis, so that cannot be the possibility as well. So diagram shows us a cell during mitosis. Now which description correctly identifies the cell? A plant, an animal, an animal. So first let's decipher the first part. Is this a plant cell or is this an animal cell? Yes, we've got these centrioles. Now this is only part of an animal cell, so this is wrong, this is wrong. Now let's look at it. An animal cell during anaphase, an animal cell during metaphase. If you remember my video, meta is middle, so center, the equator. Anaphase is apart, alag ho jate hain. So apart means they're going apart. So the answer was C, an animal cell during anaphase. When we have question 24, a gene codes for the production of a protein P53 that binds to damaged DNA during interphase and prevents its replication. A carcinogen in cigarette smoke mutates this gene. Which statement explains why this mutation may cause cancer? Cells with no P53 allow their damaged DNA to replicate. Why? Because it's not allowing it to function. What does P53 do? It binds the damaged DNA during interphase and prevents its replication. So if this gene codes for a protein P53, so it prevents its replication. Now that means it will allow its replication. So cells with no P53 allow their damaged DNA to replicate. Read the question carefully, understand what they're asking, and then of course you'll be able to figure it out. Then we come on to question 25. Question 25 says, what is the role of DNA polymerase? Condensation reactions produce DNA nucleotides for replication. No, doesn't make sense. Doesn't produce DNA nucleotides. It joins the DNA nucleotides to form a DNA polymer. Correct alignment of RNA. Well, RNA is not in part of it, so let's not even look at the RNA ones. Then formation of bonds to complete the sugar phosphate backbone. Yes. This is what it does. It forms the phosphodiester bond. And formation of glycosidic, I mean, that's a whole lot of beautiful story, because glycosidic bonds are not found in the DNA, it's found in carbohydrates. But somebody might do just, okay, A, B, C, D, let's do any, mini, minor, mo. But if you're going to do that, I'm sorry, you're not going to get many marks in your MCQ paper. Then we come to question number 26. Which row represents the correct features of the nitrogenous base thymine? I've done this with many of you. Thymine pairs with adenine. And what is thymine? It's a pyrimidine. And pyrimidine have a single ring structure. So has a single ring structure. If you remembered that, but then you know it can only be A or B. Then it's a purine. No. What are purines? Adenine and guanine are purines. And the U, this is the U part. This is the U here. So adenine and guanine are purines. So we know thymine is not a purine. So then we have this part. Then we come on to, let's now look at the third thing, forms two hydrogen bonds. Now we all know that adenine and thymine form two hydrogen bonds. And I told you C and G form three. C is the third letter of the alphabet. So C and G forms three hydrogen bonds. Then pairs with cytosine. Now that was wrong. So I'm sure everybody could do this question. This wasn't a very difficult question. Question 27. Mendelssohn and Stahl investigated DNA in bacteria. They grew bacteria in a medium with only heavy nitrogen until all the bacteria was heavy. So we've got this heavy nitrogen. N15, I'm covering it in yellow. These bacteria move from heavy to in a medium with only light nitrogen. So then what will happen? The heavy will separate out and we will get a light nitrogen, which I'm coloring in red, right? A sample of bacteria was collected from the second generation that DNA analyzed. Hybrid DNA contains both heavy and light, so this would be hybrid DNA. Which row suggests the percentage of heavy DNA strands and the percentage of hybrid DNA strands? Percentage of heavy DNA strands and the percentage of hybrid DNA molecules in the second generation produced in the medium containing light nitrogen. It said which row shows the percentage of heavy DNA strands and the percentage of hybrid DNA. Now you can see this line. This is the second generation. Now you can see this is hybrid DNA. So percentage of hybrid DNA was 2 out of 4, which is 50%. Again, I'm telling you, the percentage of hybrid DNA was 2 out of 4. And the percentage of heavy DNA was 25. Why? Because we've got heavy. How many heavy do we have? Just look at this now carefully. So the heavy DNA was only two yellow. Only these two yellow. I'm going to give it another color. Only this one. So 2 out of 8 means 1 out of 4 means 25%. So please understand this question. Why was it 25% and 50%? Percentage of heavy DNA strands. Percentage of hybrid DNA molecules. Now this is very different English. Percentage of heavy DNA strands, which are the ones I've shown in yellow. And percentage of hybrid DNA molecules. So one red and one yellow. And one yellow. This is a hybrid DNA molecules. So I hope this is clear to all of you now. This is a very long table. It says the table shows the DNA triplet codes for some amino acids. So amino acid, DNA triplet code, amino acid, DNA triplet code. So we've got 4 headings: amino acid, DNA triplet code, then amino acid, and then DNA triplet code. Now the base sequence on the DNA strand coding for a polypeptide is shown. So we have these, and they put these in three adjacent ones together, so you can see them. They've done this for a certain purpose. Now it says 2 mutations occur in the sequence during DNA replication. Which mutated DNA would have no effect on the polypeptide synthesis? You know, if the third nucleotide changes, at times that does not cause a change in the amino acid sequence. So 2 mutations occur in this. So you can see here, let's look at each one now separately. This is CCA. Now this is also CCA. And then it's TTC. This is also TTC. And then it is, here also it is TTC. Then let's look at the next one, ACG. Now this is ACA. Now that doesn't want to change. Then GCG becomes GCA. Then TTA remains TTA. And then GCA remains GCA. So it says 2 mutations occur in this sequence during DNA replication. So 2 mutations was only in B, and so the answer was B. The rest of them have more, or they have none in them. Then we come on to question number 29. A group of students were asked to look at the photomicrograph of a cross-section of unfamiliar material and make observations. The student described X as circular, a hollow tube, a spherical structure. Look at the question again. A group of students asked to look at the photomicrograph. Photomicrograph. The student described X as this. Which descriptions are correct? Why? Because you see, you can't see it's hollow. It's not a 3D diagram. Photomicrographs are just picture of slides. So you could only see it circle. You couldn't figure it out that they were hollow tubes, or you couldn't figure out the 3D structure and saying that's like a ball or a rugby ball or a football or a cricket ball or a ping pong ball. So hollow tube and spherical was not very right, and that's why one only is the answer. Then coming to question number 30, irrigating crop plants with water containing low concentration of salt causes an increase in the concentration of salt in the soil. So first you have crop plants and then you put water containing low concentration of salts, causing an increase in the concentration of salt in the soil. What explains why the increase in salt concentration eventually kills the crop? Question has to be read in a very important concept has to be clear. It said crop plants with water containing low concentration of salt causes an increase in the concentration of salt in the soil. Now, what explain why the increase in salt concentration eventually kills the crop? Because something has to kill it. The kill the crop is something which you have to emphasize on, and you have to figure this out in the question. So why is the answer C? Because the water potential in the soil decreases. That's fine. Because there's too much salt in the soil. Because it said that the concentration of salt in the soil increases. So water potential in the soil decreases and the direction of water movement is out of the roots. So water starts to move out of the roots, and this will kill the plant. It's not movement of water into the roots or the movement of water into the soil solution. No. This is not going to, we have to find out a reason why it is going to kill the crop eventually. So reading the question, understanding it, and then answering it is of course the kid who's going to be the winner. So this was question 30. Now we come on to question number 31. Now let's look at question number 31. The photomicrograph shows a transverse section through part of a plant root. Which tissue inhibits the path of water through the apoplast pathway? Now you know that that is the endodermis, which contains cells lined with suberin, and that forms the Casparian strip. So you know that endodermis is this outer layer, these cells which are the endodermis cells. So endodermis, and then we have the pericycle, and then we have the xylem and the phloem. So of course the answer was B, which is the endodermis. Then it says the table shows some information about three blood vessels, P, Q, and R from a mammal. Diameter, wall thickness, percentage composition of the wall, muscle, collagen, elastin. Which row identifies blood vessels P, Q, and R? So P has a larger diameter, Q has a smaller, and R, though it's very small, 4 millimeter. Now, what could be P, what could be Q, and what could be R? So, what are the other things? Muscle. In R, there's a lot of muscle. So R has to be, I mean, I would have thought of that. I would have thought in those lines. I would have thought R has got the most muscle. So R has to be an artery. And then you were only left with this choice, which is A. The rest of all was wrong. So more muscle was the clue in it. That more muscles is always would be an artery, couldn't be anything else. And that would have you figured that out in that way. And coming on to question 33. Which row correctly identifies the locations in which a type of molecule or cell is present? So type of molecule or cell: antigen, glucose, lymphocyte, neutrophil, blood, lymph, tissue, fluid. So we know the type of molecule or cell: antigen, blood, lymph, tissue, fluid. So all three of them would have antigens. This is how we had to do this question. Next, we come on to question number 34. The graph shows the percentage saturation of hemoglobin with oxygen at different partial pressures of oxygen. Which range of partial pressures of oxygen result in the greatest change of percentage saturation of hemoglobin per kilopascals? Which range of partial pressures? Is it 0 to 5, 5 to 10, 10 to 15, 15 to 20? Is the greatest change of percentage saturation of hemoglobin? So of course the answer to that was B. Then how many layers of cell surface membrane separate an oxygen molecule in the air space of an alveolus from the nearest hemoglobin molecule? And that of course is 5. And we've done that in another paper as well. You see this is the alveolar wall. Then this is the capillary wall. And then this is the red blood cell. So how many layers of cell membrane? Number 1, 2, 3, 4, and then 5. And once it enters the red blood cell, of course, it forms oxyhemoglobin. So how many layers of cell membrane separate an oxygen molecule in the air space of an alveolus? So this is the alveolar wall. And this is made of this one cell thick layer. And then we have the one cell thick layer of the capillary wall. And then of course there's a capillary wall here as well. And this is of course all the capillary wall. So this is the capillary. And this is the alveolar wall made up of these cells. And this is the alveolus. And then it says oxygen is inside here. So oxygen has to cross number 1. First this membrane, then this membrane, then this membrane, then this membrane, and finally this membrane. So it has to cross 5 membranes. Next, question number 36. Which biochemical is formed and what happens to the oxygen dissociation curve of adult hemoglobin when carbon dioxide combines with hemoglobin? So the biochemical formed is carbaminohemoglobin. Carbaminohemoglobin, and more the carbon dioxide, more will it shift to the right, and that's called the Bohr shift or the Bohr effect. So the oxygen dissociation curve will shift to the right. Then coming to question number 37. The diagram has been used to identify 4 infectious diseases. Infectious disease 1 is waterborne, not waterborne. Then not waterborne can be a virus, airborne, not airborne, and not a virus. Which row correctly identifies the infectious diseases? So 1 waterborne is cholera, 2 measles is airborne, 3 HIV/AIDS is not airborne, and 4 TB is not a virus. It is a bacteria which causes tuberculosis. So this is how we had to do question number 37. Then we come on to question number 38. How does penicillin is an antibiotic, affect bacteria? It inhibits DNA replication by binding to nucleotides. It inhibits translation by preventing tRNA binding to ribosomes. It is a competitive inhibitor of an enzyme in cell wall synthesis. It is a competitor of an enzyme in protein synthesis. So yes, we know that it prevents the cross-links between the peptidoglycan when the cell wall is being formed, when the bacteria divides by binary fission and new cell walls have to be formed. Now let's read question 39 slowly and understanding it. Growth factors are chemicals that stimulate a cell to divide. Growth factors are transported around the body in the blood, and they attach to cells that have complementary growth factor receptors to their surface. Some cells have too many growth factor receptors on their surface, and so the cells continue to divide, forming a tumor. Trastuzumab is used as a treatment for some cancers. You want to destroy the cancer cells. Trastuzumab works by binding to a specific growth factor receptor on a tumor cell. This stops the cell dividing and the cell dies. What is trastuzumab? Now, if you remember the monoclonal antibody, the syllabus says you've got to know it in diagnosis and treatment. And what we do is that we have antibodies and then we add a drug to it. And then we'll attach to the cancer cell and destroy that cancer cell. So a monoclonal antibody was the answer to that. But you had to understand the question to really figure this out, that it was a monoclonal antibody. Coming to the last question, what describes artificial active immunity? Artificial active immunity is when we give weakened viruses, like when we give a vaccine. Active, why active? Because your lymphocytes perform the activity. I always teach this to you like this. I say your lymphocytes have to do that activity. So that is called active immunity. If it's natural, you have the disease, you suffer, you have 10 days of fever and you're sick, and then you're okay. So you have natural active immunity. What is artificial active immunity? When we give you a vaccine, when we give you dead or weakened viruses or bacteria or pathogens, and then your lymphocytes think that's the live virus or the live bacteria, and then they start to work on it, and plasma B cells form, multiply, memory B cells form. So you do not get the disease when the actual virus enters your body. So stimulation of lymphocytes by antigens contained in a vaccine, and this was the answer to, this was the C was the answer. Protection against a pathogen by an injection of antibodies, and now that is not, there's no antigenic exposure. That is passive immunity, and that is artificial passive. Then protection against a pathogen by drinking colostrum containing antibodies. Now, of course, that is again natural passive immunity because you're getting ready-made antibodies. And then stimulation of lymphocyte by antigens on the surface of invading pathogens is like person suffering from measles and becoming sick and having fever and then never having measles ever in his life. Now that is natural active immunity. They were asking for artificial active immunity. So all four were in the answer, and you have to figure that out. That ends this paper, and thank you very much. And inshallah, another paper in a few days to come. And I hope this is all helping you all in your preparation for the paper next week on the 10th of uh, on the 10th of next month. Thank you very much.